So, welcome everyone. Today we're going to be talking about Iboga as a potential cancer therapy. I just want to be very clear: we're not saying Iboga cures cancer or Ibogaine cures cancer. Uh, what we're talking about is the theoretical application of how it potentially could, based off the research we know around cancer, around sigma receptors, and around the affinity of Ibogaine. And the central tenet, which we're going to be exploring here, is that because Ibogaine binds to the sigma receptors, and because these sigma receptors have a very important responsibility, you could say, for the survival of cancer cells, that if Ibogaine can bind to them, then the sigma receptors will no longer be able to support the cancer cells the way they do, which would lead to the death of the cancer cells. And so this isn't medical advice, not at all. This is just us exploring research. It's for educational purposes only. So yeah, I just want to be very clear. If you have any health conditions going on, not to take this medical advice, just to go and see a doctor. You know how that story goes. So yeah, we'll be exploring a little bit of introduction to cancer. We'll talk about some of the sigma receptors. What's their role with cancer specifically? We touched into that earlier, just a second ago. Ibogaine and sigma receptors, and then we'll talk about the therapeutic potential and sort of wrap it all together as one. The first thing that's most important to talk about is the prevalence, right? Like why are we doing this? Why are we talking about this? And the reason is because we're expected to have over 2 million new cases of cancer in 2025. That's over 2 million families that are going to be affected by this. So 2 million people that are going to have to hear this news in some way. That's about 5,600 cases a day. So you hear that number, it's shocking actually. Over 600,000 deaths in 2025 will be the projection. That's 1,700 people dying a day. Yeah, it's tough right there. And in women, the most presenting form of cancer is breast, and in men it is prostate by far, actually. This is a chart I found from Our World and Data. It's showing the cancer deaths versus the crude cancer death rate versus the age standardized cancer death rate. We'll start from the bottom. The age standardized death rate is essentially showing it's decreased by 20%. And the reason that's happening is because what this is really showing is that our therapy options for keeping people alive are improving. So people are actually living longer. Less deaths from cancer. However, that's based off age. Now, if we just take the number as it is, you see at the top, there's a red, the cancer deaths. So that's a sky high number. And then the crude is the cancer deaths over 100,000 people. And again, it's on the rise. But basically what's happening is there's so many more people being born, there's so many more cancer rates showing up that, the numbers looks like it's going to go vertical soon, which is scary. So what is cancer? Cancer is a group of diseases characterized by uncontrolled growth of the cell and the spread of abnormal cells throughout the body. And the result of this is it has a downstream effect, as we all know. I'm sure if anyone listened to this, you probably know somebody who's affected by cancer. I mean, best friend his mother passed from it. I had a best friend whose mother passed from it years and years ago. It's very prevalent everywhere. And so what is this uncontrolled cell growth about, right? Cell growth and division is a normal process in the body. It's our cells are doing it all the time. And our cells receive signals to start dividing, and they get signals to stop, right? This is how we replace old and damaged cells. This maintains healthy balance in the body, which we call homeostasis. However, in cancer, the cells will divide uncontrollably, even when they're told to stop. And when the triggers go off for apoptosis, which is programmed cell death, the triggers are actually often disabled, allowing cancer to persist. And this is due to ER stress, which stands for endoplasmic reticulum stress, which we'll talk about for a little bit. And the result of this is tumor form. So it's sort of the mechanism in a nutshell. So the question then becomes, what is causing these different what's causing this ability for the cells to continue to grow? The first is genetic mutations. You could easily throw environmental toxins into that category. With all the pesticide and all the information what's going on in foods, it's pretty clear what's happening. Sustained growth signals. So cancer cells or normal cells require signals, external signals, in order to get them to divide. Imagine like the call from the commanding officer saying, "Hey, time to start dividing." And what's different about cancer cells is they sort of just do it on their own. Evading apoptosis, that's the programmed cell death. And so again, cancer cells have an ability to evade the program to die. And so we'll talk about that a little later. And then the same way they don't wait to receive signals to divide, they also ignore the signals to stop dividing, right? And we have immortalization. This is a very, very interesting one, topic of a lot of research. But basically, we have these things called telomeres, and they allow our cells to only divide a certain amount of times, right? Because the telomeres shorten every time. What's so fascinating is that cancer cells have this ability to activate telomerase, which extends the telomeres, allowing them to essentially become immortal. Angiogenesis, this is when tumors grow, they start to require more oxygen. Just like if you went for a run, you would start breathing heavy, you'd require more oxygen. As these cancer cells proliferate and grow, they require more oxygen. Metastasis, this is when they basically move on to nearby areas. This is one of the main issues we find with cancers, especially in the brain. And then the evasion of the immune system, of course. They develop strategies to evade detection. And all of this results in a sort of stress on the internal system, right? Just like if you were lifting very heavy weights, for a simple example, there's a lot of stress and strain on the system. The same thing for the cancer cells, so constantly proliferating, growing, and expanding. That's a lot of stress there. And so the question is, how do they manage that stress? So when this stress occurs, it predominantly happens within the endoplasmic reticulum. I'll show a cut slide of a cell in a second, but the endoplasmic reticulum is like a factory for delivery, or a delivery system inside of your body. It has a few different functions, proteins it makes, fats it makes, and the folding and fixing of these proteins. And it stores and controls calcium. And we'll talk about the reason why this is so important, but just remember that calcium part for now. And it experiences stress when it's overwhelmed and unable to handle its functions properly. And so there's a few things that might cause that stress: protein misfolding, calcium dysregulation, ROS, reactive oxygen species, which results in oxidative stress, lipid metabolism imbalances, cytokinesis, inflammatory versions, hypoxia, lack of oxygen, toxic and drugs, and viral infections. And so what you're seeing here on the right is a picture of the endoplasmic reticulum under stress. Don't worry about the cascading effects down there. We'll talk about all this later. But for now, what's important to note is that in order to handle that stress, what it uses are these things called sigma receptors. Yeah, there's one right here, right here, right here. And as you're going to see, these sigma receptors act as chaperone proteins in the endoplasmic reticulum, and they regulate the stress response, signaling pathways, and maintenance, the cellular maintenance, the homeostasis of the cell. So they're responsible for that. So just to take a step back, if we're saying cancer cells are growing a whole bunch and that they're under a lot of stress from this growth, then what we find is that the sigma receptors are responsible for handling that stress. Right. And if that stress can be handled, the cancer cells can continue to survive. If the stress can't be handled, now the cancer cells cannot continue to survive and they'll die. And so as you're going to see here in a moment, what we're really talking about with Ibogaine's potential is its ability to bind to the sigma receptors and stop it from handling the stress that's going on in the cancer cells. So there's two types of sigma receptors. And actually, before I say that, I should mention that they show up in a few different spots. They show up in the rough endoplasmic reticulum of the cell, which is the majority of it here. And the smooth, they show up in the plasma membrane and also on this, in the mitochondria, specifically on a place called the mitochondria-associated membranes. They're called MAMs, excuse me. But just think about them, about just how they show up on this outer membrane. They show up on the membrane around the mitochondria as well. And we'll talk about this in depth. There's two types. There's sigma 1 and sigma 2. Sigma 1s are these chaperones, really. And Sigma 2s are very vital in the cell handling responsibility of the stress. And so we'll talk about that here in a moment. We'll go deep into that actually. A little difference between Sigma 2, we're not going to go over all of this, but what's important to mention is that in Sigma 2 receptors, they're recently identified and they're linked to these TMEM97. It's a gene and it's a transmembrane protein 97. And we'll see why that's important later. So I just want to name that. And then they're also involved in lipid metabolism, and cholesterol regulation, which is going to be primary to our theoretical application here as well. The next section here, we'll talk about what is the actual role of the sigma receptor in certain types of cancers like breast cancer. So this is a white paper. If you want to find the DOI on it, it's down at the bottom. That means you can just type that in and you can read the paper for yourself. I'm a blast. But the role of sigma 1 in breast cancer is what this paper is about. And what it's essentially saying is that in especially one of the most aggressive types of breast cancer, which is called the triple negative breast cancer or TNBC. And what's so special about this one is there's certain receptors in this one, estrogen and a few others, that don't show up here in this type of breast cancer. And the reason that sucks is because the treatments for this specific type require binding to those receptors that don't show up, so it makes it almost untreatable. So it's a very aggressive form, and it's sigma 1 is overexpressed in these types. And that correlates, if there's a lot of sigma 1, then what we find out is there's lower patient outcomes and poor survival in breast cancer patients. And so the question is, why would they be overexpressed, right? If we remember that it's involved in managing stress, these sigma receptors. And so if there's a cell that's overstressed, it makes sense that there'd be a lot of stress reducers there, right? And so on the next slide, we'll talk about sigma 2 receptors. This page talks about them being sort of the gatekeepers. News, they're basically finding that these sigma 2s are a new source of focus for fighting against cancer. The overexpression in tumors, again, highly expressed in several cancers, including breast and lung. Excuse me. And then what's the mechanism of action? The sigma 2 receptors specifically help cancer cells manage endoplasmic reticulum stress. And they do this by regulating the intracellular calcium signaling between the endoplasmic reticulum and the mitochondria. So if we just go back to our slide real quick, we have the endoplasmic reticulum here, and then we have the mitochondria. And so what we're saying with sigma 2s is they do a masterful job. Part of their function is regulating the calcium exchange between the two. So the question we need to ask is, well, why is that important? Right? Like, what does that mean? Like, who cares? Doesn't seem like a big deal. The reason this is important is because there's a few things that the cancer cells require or rely on in order to reduce their stress. The first thing they rely on is that the proteins within them are folded correctly. Yeah. And so what happens is, in order for these proteins to fold correctly, they require calcium-dependent chaperones. And these ensure the proper folding, which then reduce endoplasmic reticulum stress. I'm not sure if I mentioned this already, but in a previous lecture we gave on Parkinson's, one of the primary causes for the degeneration of the dopaminergic neurons in Parkinson's is that these proteins fold incorrectly. And when they fold incorrectly, they can't be broken down or removed. And so they start to aggregate inside of the space around the neurons, creating a toxic environment. And this causes death to that neuron, which means no dopamine is produced and have a cascading effect, which we have the Parkinson's. Very similar thing here, right? So the cancer cells are overworking. They need this large amount of proteins to be folded correctly, and calcium is reliant on that, right? And that's what sigma 2 receptor does for the cancer cell. It keeps the calcium exchange happening. ATP production. The mitochondria uses endoplasmic reticulum-derived calcium to activate the tricarboxylic acid cycle and generate ATP. So if you remember anything from high school biology, ATP is produced in the mitochondria. These are the energy units our body uses. Plants do it too. They take ADP and convert it to ATP. And so basically there's the energy units. So the mitochondria itself requires on calcium to activate the cycles to produce energy. Apoptosis, this is the programmed cell death prevention. Proper calcium transfer prevents endoplasmic reticulum stress. And endoplasmic reticulum stress is what triggers apoptosis, cellular death. And so the reduction of that stress will stop the apoptosis from happening, right? So again, why is this important for cancer cells? To make sure they have calcium exchange so there's no cell death triggered. Another reason is for ROS regulation, and this reacts to oxygen species. ROS is, you could think of it like a spark, is the easy way to say it. ROS is like a spark, and if you think about a spark, a wildfire can be started with a spark, and so could a small fire to keep you warm at night. So it's not a bad thing necessarily, but too much of it is definitely a problem. So this is why it's important for cancer cells to have these sigma-2 receptors, because they help handle all of these vital functions for it. And now you see why they're overexpressed in cancer cells, because cancer cells need a lot of maintenance and upkeep to keep going. And so let's do a short recap, and then I'll sort of present the tenon again, and that way you can keep tracking. So what we know is that there's sigma-1 and sigma-2 receptors, that they're both expressed in cancer. They act as managers that help handle stress inside the cancer cells, mainly in the endoplasmic reticulum. We know that the SR1s and SR2s work in different spots, but a big spot they work is between the mitochondria and the endoplasmic reticulum. And so the question we need to ask ourselves is, okay, if we're curious about the effects of Ibogaine on the sigma receptors in this treatment for cancer, then does Ibogaine even bind to the sigma receptors? If it does, what would that binding possibly do? And you're sort of starting to see the therapeutic potential, right? That if you have these sigma receptors that are so responsible for maintaining a state of healthiness or liveliness inside of the cancer cells, well, what would happen if those receptors were now bound and they couldn't do what they are meant to do? What if they couldn't move the calcium? Well, then the cancer cell would be in trouble. And so you're starting to see the therapeutic potential here of Iboga and Ibogaine. So the first question we need to ask is, does Ibogaine bind to the sigma receptors? And before I get to there, if anyone doesn't know what Iboga is, it's a plant that grows in Africa, a perennial shrub, native to Central Africa, Gabon, the Congo, the Democratic Republic of Congo, whatever you want to call it, and Cameroon. The Bwiti people are the indigenous people there who hold Iboga, who are the trusted bearers of it. I share this sometimes, but my father, who I've never met, I took a genetics test, however, has lineage back from Gabon. My first time ever working with Iboga, after that ceremony, when I got back to the States, I took a genetics test and found out that my father was from Gabon. That's sort of what catalyzed me in this work, in a way, you could say. But Iboga is the tree bark itself. And then out of Iboga comes Ibogaine, a single alkaloid that people like to extract. The way I sort of frame this, when people ask what's the difference between Ibogaine and Iboga, the way I say they're like, what's the difference, right? Was one better than the other? Ibogaine is obviously the most researched. It has tons of effects, psychoactive and neurobiological effects. Studied a lot for addiction and PTSD treatment. But the way I sort of frame it, and I say, imagine if you had a canvas and I would tell you to paint something for me, but I just gave you the color red. That's like working with Ibogaine. Now the next person B, the same thing, give them a blank canvas and they're going to go paint. And now I give this person the entire rainbow of colors. That's what Iboga is. There's about 12 to 14 alkaloids inside of Iboga. And so you're talking about, just a broader range of therapeutic potential, right? And also Ibogaine has certain cardiotoxic effects, which we'll talk about a little bit. And the QR code down here is if you're interested in giving back to the Bwiti people at all, if Iboga's touched your life and something you feel called to do, of course, no pressure. my wife and I have a nonprofit called Sacred Roots Foundation, and we're always open to receiving donations. All the money goes directly back to the bone. We fund 100% of the maintenance of the nonprofit through from our own personal income, and all of the donations go to they go directly to the village restoration projects we're doing. So I just want to name that real quick. And back to our presentation. So here's another white paper again. The DOI is down here. The DOI, the DUI. And this one is talking about the connection between Ibogaine and sigma receptors, right? Because we're asking, like, do they even bind? And the answer to here is clearly yes, they do bind. This Ki number, the lower is the better. This basically just represents the affinity in nanomoles. And so the lower the number, the better, right? And when we say affinity, we're talking about sort of like a lock and key. How well does it fit, and how attracted really is it to those certain receptors? And so it has a moderate affinity, Ibogaine does, for the sigma-2 receptors, and good selectivity, way better over the sigma-1 receptor, which is 8,554 nanomoles, which, remember, the lower the number, the better. And so what they're saying here is it's only, and we talked about this in other presentations, that Ibogaine specifically binds to multiple receptors in the body: the NMDA, the opiate, the serotonin, the dopamine, the sigma, the acetylcholine, that's for nicotine. So it has this very wide range of benefits, which is why Ibogaine is so helpful. But when you, again, apply that with the other alkaloids, you're talking about a whole different beast. And then this paper looked into developing derivatives inspired by Ibogaine. So other compounds with similar backbone or shape that could also bind to these SR2 receptors, right? And so it's sort of like taking what nature made and trying to build and piggyback off that for specific ones that have a higher affinity. So we answer the question, yes. Does Ibogaine bind to the sigma receptors? Yes. So then the question is, okay, if it binds, and what is the potential that could happen? And so how would this help with stopping cancer, really? So the first one you talk about is the cell proliferation and inhibition. So Ibogaine has a high affinity for sigma-2, which we talked about, which are abundant in tumor cells, which we talked about. And therefore, this could disrupt the signaling pathway and inhibit cancer cell proliferation, right? And again, if we're saying that these sigma receptors are responsible for managing the stress of cancer cells, right? These cells are overworked, they're growing and growing, they need to manage their stress, and the sigma receptor is responsible for that. And the sigma receptors require things like calcium to bind. Then what happens if that sigma receptor can't bind calcium because Ibogaine's in its parking spot? Well, then the stress won't be managed, and the cell would essentially die. The cancer cell would die off. And so this is what we're talking about when we talk about Iboga as a potential therapeutic approach. So this paper on the left is basically just saying one of the ways it will do that is by disrupting the endoplasmic reticulum stress responses of the sigma receptors, as we just talked about. And it would do this by regulating the calcium, the intracellular calcium, and that might make cancer cells more susceptible to apoptosis, right? No calcium exchange can happen. So remember all those things calcium is responsible for in the cell, and so no calcium exchange is happening. So now you have this cascading effect, which makes the cancer cells at a greater risk of death. The other paper is talking about how an affinity for the sigma-2 receptor suggests that it can effectively interact with the transmember 97 protein. We introduced this earlier. And why is that transmember protein critical? It's because it influences the activity and cholesterol transport and metabolism. And we'll talk about that one a little bit more right now first, and we'll come back to the calcium one in a moment. So for people who don't know, there's this really weird thing in our food sort of language and culture that cholesterol is this bad thing. Now, too much of anything is bad. Like too much of water is bad, too much alcohol, too much Iboga, too much anything. It's not healthy for you, right? Like, let's just be honest about it. So the thing with cholesterol is we have to understand that the myelination around our neurons is cholesterol. The white matter in the brain is from cholesterols. And inside of your cells, the membranes themselves, the integrity of it is supported by cholesterol. You can think of them as like the support beams inside of a structure of a building, of a home. If you look around your home, if maybe you have the woods exposed, if they're not, you have support structures all through the house in which you're living in. And without those structures, the house would crumble. And the role of cholesterol is not much different, right? And so what we're saying here is that if Ibogaine has an affinity for the sigma receptor, and the sigma receptor is correlated to the transmembrane 97 protein, or enzyme, excuse me, which is responsible for cholesterol trafficking, right? That transmembrane protein is responsible for the trafficking of cholesterol, placing it where it needs to be. Then by binding to that, Ibogaine could potentially destabilize the cancer cell's membrane and interfere with their signaling pathway. And so if you look down here at the bottom, there's a really good image. Now, this is talking about hot and cold, but don't worry about that right now. Just imagine that without cancer, these proteins become, there's no integrity, they lose their fluidity, and there's no cellular signaling either, right, without cholesterol, because the reason these membranes build, they build this thing called the lipid raft up top. And this is where proteins are sorted, actually. And so if this integrity of this entire thing isn't right, it's going to collapse, just like a building. The same thing would happen with the membranes of the protein. And if you ever worked in a chemistry lab or used alcohol to sanitize your hands, the reason why you use alcohol specifically is because you're creating cellular lysis. And what you're doing with that is you're using a strong alcohol, and it's essentially eating through the cell wall of the bacteria, the single cell organisms, or the multicellular organisms, or whatever. You're basically viruses, you're destroying the cell wall of them, and that causes them to die. And the same thing here. So what they're saying is Iboga has a potential. What we're saying is Iboga has a potential to bind to the sigma receptor, stopping the ability for the cholesterol to be trafficked, which would inhibit the integrity of the cellular membrane of the cell, right? And so that's what we say at the bottom. Without cholesterol, the cell membrane would become too rigid. And we say in hot temperatures, and brittle and prone to breaking, cold temperatures. Yeah. And so you're starting to see kind of where we're getting at here with this. So that's one layer to this. The next one is apoptosis induction, right? Apoptosis is programmed cell death. And so what we're getting at, the theoretical approach we're having here is if Iboga or Ibogaine can bind to the sigma-2 receptors, this could trigger apoptosis, disrupting mitochondria function, and not allowing the endoplasmic reticulum stress to be handled. And so there's a few ways in which this could happen. The mitochondrial membrane permeation, we'll talk about that. The endoplasmic reticulum stress, we've explored that in detail. The calcium dysregulation, we'll talk about that in much more detail now. And then it also has a selective cancer cell targeting. And what we're saying here is that because sigma-2 receptors are overexpressed in cancer cells, again, this is because they require them to be there, it makes them perfect targets, right? If you're looking to target a bunch of ducks in a pond and there's 100 ducks together, you're going to have a better chance of hitting them, right? Because you know where to aim, versus if there's one duck here and one duck there and one duck somewhere else, you won't really be able to spot them. But when there's 100 ducks together, you start to see, okay, and now you know where all the Ibogaine is going to go. So it sort of works as a selective targeting, because that's what the cluster of receptors are there that they have an affinity for, right? And so we'll talk about these and break them down in some more detail here. And so the first one is this endoplasmic reticulum stress. We talked about this a few times, but it's important that we sort of walk through this so we can understand the potential of Ibogaine, right? We can't just say, oh, Ibogaine binds this receptor and, has a potential cure for cancer. Like, let's talk about why and how, so we really deeply understand this. So we'll start step by step. So the sigma receptor is there. We introduce Ibogaine to the system through Iboga, through pure Ibogaine, whatever it could possibly be. Again, this is all theoretically, so information covers only. Ibogaine would in theory bind to the sigma-2 receptor because it has an affinity for it, which we showed. From that, the endoplasmic reticulum stress, which is being managed by that sigma receptor, would no longer be managed by it, which means the stress would amplify. Yeah, the stress would go up. They're already under stress, like we said. Because the stress would go up, right here, what would happen is the protein folding would be inhibited. And remember, we talked about the protein folding has to be done right, or else they start to coagulate. They start to aggregate inside of the mitochondria, inside of the endoplasmic reticulum, and it's going to cause all sorts of problems, just like it does in Parkinson's. And then what we find is the endoplasmic reticulum proteins become misfolded, so they aggregate up. So now the cell starts producing more chaperone proteins to try to help to catch up with the unfolded proteins and to repair the ones that are misfolded, right? And these are all collecting in the endoplasmic reticulum. I should have put a space in here. My apologies. Activates, and then what this is going to do when they start to produce these chaperone proteins, it's going to activate something called the unfolded protein response, or UPR. This is a stress management system that we have. It's fascinating how fucking intelligent our bodies are. It's insane. Like whoever or however this was built is good job. The UPR is a stress management system. Yeah. And what this UPR does is right here, you see this UPR activation. If the new protein production can be sort of taken care of, if all these aggregating proteins, these misfolded ones, can be fixed and folded correctly, then what happens is we find a state of homeostasis again, right? We can find this sort of balance, and the cancer cell could live on and keep going. This is the normal pathway that's happening. But with Ibogaine binding, hopefully what could happen is the stress increases, the proteins aggregate, and the UPR cannot keep up with it. And when it can't keep up with it, it causes further and further stress. This sends a distress signal to the mitochondria. To a certain point, it crosses the threshold and it says, hey, mitochondria, we have a problem. What's going to happen is that mitochondria is going to cause calcium leaks and oxidative stress. And this leads to mitochondria dysfunction, leading to the cytochrome C release and apoptosis. We'll talk about that part later. But if what we're really saying is by inducing more stress, the proteins will build up. When the proteins build up, will cause the mitochondria to essentially lose its calcium and leak and break. That's almost like ripping the engine out of a car, right? You're basically killing the cell from the inside out. So there's one potential approach on how Ibogaine could, in theory, assist in cellular apoptosis by causing more proteins to aggregate. Proteins aggregate, it activates a response. If the response can't handle that, it's going to cause the mitochondria in the cell to die by leaking calcium. So now let's talk about this calcium dysregulation, because that's kind of where we got to. And so remember, we have this now, we're at this point of mitochondria dysfunction. And so first we'll talk about this chart on the right. I think it's easier to start with the visual. So up here on this left, on the y-axis, we have ATP output. On the x-axis, we have the calcium load. So what we're showing here as the calcium starts to load, starts to increase, the ATP peaks, and there's a certain point where we have this maximum yield of calcium activation, right? And you can consider this like the ultimate point of health, right? And so once calcium starts to overload, though, we see that the ATP production starts to taper off, and then when it gets to this zone back here, it has this very sharp drop off, yeah? Where there's no more ATP production. No ATP means no energy for the cell to use, which means it will die, cannot survive. So now that we understand sort of what's happening from the ATP standpoint, imagine Iboga binding. I begin binding to the sigma-2 receptors. As we remember, the sigma-2s are responsible for the calcium flow and maintaining calcium homeostasis. So it's going to block the calcium flow, right? This causes mitochondria stress and dysfunction. This interferes with the ATP process. This is what we're talking about over here, the ATP process being interfered with. This is going to disrupt the entire electron transport chains, making energy creation insufficient and almost impossible to a degree. Excuse me. As this happens, oxidative stress will start to increase inside the cell. This results in overloads to higher level of the ROS, which damages the mitochondria membrane and proteins, right? If we go back to the slide on this, what we're talking about, that this membrane gets damaged due to the oxygen species. Remember, they're like sparks, they're like wildfires. So imagine them sort of just burning through that is a simple analogy to try to hold. This damages the mitochondrial membranes and the proteins inside. The mitochondrial permeability transition happens. The mitochondria becomes too permeable, causing them to swell, leak, cytochrome C and lose function. And the result of that is the ATP depletion, oxidative stress, and cytochrome C release triggers apoptosis. We'll talk about cytochrome C in a second. But what's important to remember is in the first cascading effect, we have the proteins aggregating, which is causing the mitochondria to have dysfunction. And in the second aggregate effect, we have the reduction of calcium inside of the system, which ends up leading to mitochondria dysfunction and cellular death. Right. So there's two different mechanisms at play here. And the next one is with the mitochondrial membrane premineralization and how Iboga might trigger this apoptosis cycle. So we'll explore that a little bit. So if you remember here, we got to a point where we said, hey, what's really happening is the mitochondria is too permeable. It causes them to swell and to leak cytochrome C and lose function. So what is this cytochrome C we're talking about is what we're getting at here. So again, we have that cellular binding to the sigma receptors. We have the disruption of calcium like we talked about. Now we're going to take it on now from the cytochrome C part. So what happens when the mitochondria swells and leaks, it's going to produce something called cytochrome C. Cytochrome C is an enzyme. Once it's released into the cytoplasm, it binds to this APAF1. This interaction forms a structure called apoptosome. I can never pronounce this word. My wife and I were laughing about this earlier, so good luck with that one. Which activates this Caspase-9 reaction. Yeah, Caspase-9 then activates Caspase-3. Caspase-3 is set to 7, which executes the cellular breakdown and programmed death. And so what we're seeing here, what we're saying is this, the potential is that by Ibogaine binding to the sigma receptor and disrupting the calcium process, you force the cytochrome C to be produced once the mitochondria swells and leaks. This mitochondria creates this molecule, which I can't say, this enzyme. And that is going to create apoptosis. That's the signal for apoptosis, programmed cellular death. And again, we talk about the selective targeting because the sigma receptors are overexpressed inside of these cancer cells. And so if you have something overexpressed there and you have something that's binding to these receptors, you're no longer going to allow them to perform their function. And so that is essentially the mechanisms behind it. I think that an easy way to recap it, we went into some of the science. We didn't go too deep into it, right? We sort of explored it just to have a working awareness. But one of the easiest ways I think we can hold this is just to say that in order for cancer cells to survive, they require sigma receptors to help them manage stress. And so what we're talking about, the Iboga, is potentially removing, by binding to the sigma receptors, not allowing them to support the survival of these cancer cells. That's the theoretical premise of it. And so what we need to do to explore this more? So the first one is sigma affinity of screening. And so what that means, what we really need to do is, if an experiment someone was interested in doing, if I wasn't doing what I do, I would probably be a researcher and spend my life studying Ibogaine, honestly. And doing experiments like this would be incredible, actually. Identifying which alkaloids in the entire Tabernacti Iboga plant bind to the sigma receptors with the highest affinities. They'd be really beautiful to characterize all the individual alkaloids. And then to say, hey, this one has the highest affinity, right? And I think there's ibogainamine. I think there's is the one that already I know for sure might have a higher affinity. I'm not sure. We didn't really go into that. We kept it at Ibogaine at this. The next one is the mitochondria function ATP production. It will be important to determine how Ibogaine's binding to the sigma receptors affects mitochondria function and energy and metabolism in cancer cells. That'd be a beautiful study to do. Also to do instead with Ibogaine with all the other alkaloids and run a comparative analysis to see which one has the highest effect on mitochondria function, and then to either extract it or use it as a model like that paper was talking about in order to create other substances that can connect. One of the reasons they don't want to use Ibogaine is because of the cardiotoxic effects. For those who don't know, it binds to a channel in the heart called the HERG channel. And when it binds there, it can't use potassium. It can't bind to potassium anymore. And potassium is what allows that channel to control the polarization and repolarization of your heart. And so because Ibogaine is bound there, it causes that effect on the heart. That doesn't allow potassium to bind. Because potassium doesn't bind, it prolongs your QT interval, your space between the heartbeats. When the the QT interval elongates, it lowers your blood pressure, right? If you think of the heart as a pump, it's pumping blood throughout your system every time. Well, when that slows down, the pressure drops. That's why we call it blood pressure. The pressure drops. It might not have enough pressure to make it up to the brain, and you have a stroke. That's how it happens. The thing about Iboga is that Iboga is only 8% Ibogaine in the wood bark. And so to eat enough Iboga to have the heart effects on there, it would be very difficult. You'd have to eat a lot of Iboga to do that without pre-existing issues and without other contraindicating medications, of course. So there's those factors in there. And then the other test we can do is a test on the effects of Ibogaine-induced endoplasmic reticulum stress. Right when the Ibogaine molecules bind to the signaling receptors, what is that endoplasmic reticulum stress looking like? How would we measure that? And you can do it again with all the other alkaloids. So that'd be the follow-up studies to do, just to name a few. So if there's any researchers watching this, if you want to do any of these, just send me the white paper when you're done. That'd be amazing. And then I just want to introduce this. This is the man I've been studying with for years, named Steve Montoya. This is him working on my wife. He works out of this cave in Española, New Mexico. It's freaking beautiful. It's made out of sandstone. It's hand carved. So yes, this was a mountain that someone went in and carved this entire thing. Yes, that is hard to believe, and it's real. You can see down here, there's a drum built into the floor. This is where he works out of. Again, he's working on my wife, doing a session on her. Steve is, for lack of a better term, a miracle worker, a shaman, whatever you want to call him. The point is I've sent hundreds and hundreds of people to him. He has cured cancer for so many people at this point. It's not even like, at first you're like, what, like, how does that happen? Now you just get it. And what he's actually going to do is he's going to use light. That's his gift. He has a very high frequency of light he works with. And he's going to wrap the cancer cell in sort of a womb or a cocoon of light and shrink its size down to the point where the body's able to sort of take in and take over again. He worked with a friend of mine's mom and helped remove brain tumors. I mean, it's zero BS. I mean, that filled my heart. So if you're watching this, you know anyone with cancer in the deeper states, even like stage 4, maybe some of these ones where, there's really no hope left. My best advice is reach out to someone like him. Not to say to stop doing your treatments and all that stuff, but because I've seen him do miracles, number one. Number two, I've also seen people go to him who are in the later stages, where it was too far progressed for him to save. But what he did was help them have and find acceptance in the dying process. And I think for each of us, we'll all be there at some point in our lives. The acceptance there is really the secret. We all want to have that acceptance as we transition out of this world. Nobody wants to leave this world and be in a state of, just regret and all this kind of stuff. It's going to be a really beautiful gift to give to someone. So if you want to reach out to him, you have our contact information in the chat below. He's a really beautiful man, really sweet, really kind, safe to be around alone. I've known him for years and years, and he's a miracle worker. So I just want to share that. If anyone's watching this and it speaks to you, please reach out and connect with him. Worst case scenario is, you're back to where you are, right? And just for a quick side story, the reason he works out of this cave is not his. The landowner is a doctor. She has a house on the Rio Grande River, so this is in the back across the river from her yard. Excuse me. And he went to a party years and years ago, and he saw this woman, and he could see, he sees a third eye type of seeing. It's not esoteric either. You literally have photoreceptors on your pineal gland, so he's just more attuned at seeing that, right? And seeing doesn't mean see. It's a different type of seeing. Anyways, he saw that this woman had a rare type of cancer, and he went up to her and he said, "Excuse me, ma'am, I hate to, I'm sorry, this is going to sound weird, but I just want to tell you, you might have a really rare form of cancer. I think you should go get this checked." And she actually cussed him out and kicked him out of the house. He's a friend of hers party. And so he just left, no problems. And she called him 2 weeks later and was like, "Hey, the day after you said something, it was in my head, so I went and got checked. And sure enough, I have this tumor in the spot. And how'd you know? And he explained to her, and he worked on her, and he healed her. And so she lets him use the cave. It's a true story. So yeah, amazing, amazing man. One of those people you want to meet before he transitioned out. And the last thing, yeah, I just want to close by saying that we, again, this is just therapeutic, or I mean, this is just theoretical applications again. So please don't take this as medical advice. Conduct your doctors, and yeah, do your own research too, because there's a lot of information out there about diet, alkaline diets, and things that are really just not taught. I don't think in the medical system as much, and not produced, maybe not as profiting. So yeah, just do your own homework. Look at all the resources you have out there. Again, we're not saying Iboga is a cure for cancer. It's a theoretical application for treatment, is what it is, though. And so, if you do want to chat about this or ask any questions, or yeah, just talk as people, and just feel free to reach out to us. We're always here for everybody.